Gerry McCauley from the company OmniSpirant.
Thanks, it's great to be here today. So OmniSpirant is a startup company aiming to develop bioengineered exosome advanced therapeutic medicinal products to treat a variety of respiratory diseases. We have a couple of early indications in mind, and some we're going to explore with the help of CALIN partners. So just briefly on exosomes, I'm sure you're all aware, nature's own nanocarriers that mediate cell-to-cell signaling, pretty much present in all bodily fluids and produced by all cells. And they can be used to mediate changes in cellular behavior, either naturally or forced, and repression or enhancement of gene expression. It's really an exponential field of research, and there's real transformational potential here in regenerative medicine, but also for drug and gene delivery applications. So our platform is based on a number of components. The core aspect here is the bioengineered exosomes. So we'll take some allogenic mesenchymal stem cells and use lenti-vectors with constructs to transform those into cell factories, little cell factories that produce exosomes, which contain overexpressed microRNAs, or conversely, you can overexpress anti-mirs to knock down mirs in your target cell populations. Some messenger RNAs have also been shown to transfer into exosomes produced by these cells, but it's much easier to get shorter nucleic acid sequences like microRNAs to reliably package into exosomes. So the exosomes, once they're isolated, will have these overexpressed messenger RNAs, microRNAs, or anti-mirs. It's possible then to use constructs like circular RNAs or long non-coding RNAs as sponges to downregulate your targeted microRNAs in target cell populations. So in such a manner, it gives us, the targeted delivery into specific cell types to greatly control multiple biological pathways by increasing gene expression or indeed knockdown of genes mediated by microRNAs, or else you can introduce your messenger RNA constructs, such as cystic fibrosis transmembrane conductance regulator messenger RNA, to get into your target cell and restore that expression of that protein. You can overexpress your microRNAs to downregulate genes that are overexpressed and causative in disease processes, or you can knock down, enhance or restore that gene expression where you have upregulated microRNAs in the body and by overexpressing your anti-mirs. We're targeting lung diseases, so in this manner, localized aerosol lung delivery minimizes the potential for off-target effects greatly. Fully acknowledge that some of these diseases also have systemic components. So just to have a look at this platform, what it might look like, therapeutic approach is mediated on aerosol delivery to the targeted areas of the lungs. And if we look at exosomes and the amount of cells in the lung we have to transfect, your aerosol delivery technology has to display a very high degree of biocompatibility, number one, not to damage your exosomes or their cargoes. And number two, you're going to encounter very severe natural barriers in the lung, like mucus layers. So you're going to need to send quite a lot of exosomes in high doses to those areas of the lung to be able to transfect your target cells. Getting past the mucus barrier is a well-recognized challenge as well, which we'll talk about later. So this packaging and targeting depends on your cell type of origin. We're essentially looking at using mesenchymal stem cells, adipose-derived, and for ease of handling and to facilitate large-scale commercial production, because some of the diseases we're looking at have huge patient populations to treat. We'll be looking at using immortalized cell lines. So we've got our cargoes of choice within the exosomes, and you can also influence the target cells and how they're uptaken by exosomes with a variety of surface ligands or surface modifications. So when we look at the lung, you won't get to any cell unless you can get past the mucus there. We're looking at cystic fibrosis, and that's hyperviscous mucus there. We're also looking at COPD. And if you want to affect gene therapy, you're not going to do it unless you can get these exosomes past the mucus layers. So already we've taken our exosomes, isolated by standard ultracentrifugation, and they've got quite a bit going for them in terms of the nanoscale. It's conducive to mucus penetration, but your surface charge is going to just hinder and keep these things within mucus and they'll be pretty much impermeable. So we successfully surface engineered exosomes with ideal mucus penetrating properties. So we've taken the surface charge down from a negative 16.5 MV down to about 2.6. These exosomes have hydrophilic properties, same nanoscale, and we're conducting, very soon we'll be conducting experiments, in vitro experiments, where you get cystic fibrosis epithelial cells that differentiate out in culture. They produce the same characteristic mucus there as you'll find in the lungs of CF patients, and applying these exosomes to the surface and tracking them through in real time, confocal microscopy, and seeing target cell internalization. So we'll soon have that, which is pretty exciting data, proof of concept data. Aerosol delivery, as I mentioned before, is a problem when you look at lung gene therapy. It's a long history of 20 years of attempts and pretty much failure in the context of cystic fibrosis gene therapy. We can look at what you're essentially looking at is a very concentrated nanocarrier formulation. We look at the marketed aerosol delivery technologies, so look at mesh or ultrasonic or jet nebulization. We've learned from the past that these technologies can have great difficulty in aerosolizing, say, for instance, here, liposomal plasmid DNA formulation. It was quite dense or quite viscous, more viscous than actually a couple of the types of nebulizers would allow. And you got pretty much suboptimal delivery through jet nebulization with external compressors. And that also had problems in that your liposomal DNA complexes were concentrated in the reservoir, so they weren't actually getting delivered in the aerosol as much, staying in the reservoir. So we're going to address this from an early stage with any aerosol delivery technology works. It's a formulation delivery device interface. And we're also going back to our exosomes in nanosuspension, and they should behave perhaps well enough to be able to deliver therapeutically relevant doses with some marketed technologies, but we're also looking at a surface acoustic wave technology that has the downside of being a benchtop demonstrator and not CE approved or FDA approved at the moment. So this formulation and delivery technology combination must minimize any damage to nucleic acids and also meet the therapeutically relevant aerosol and dose delivery to have therapeutic effect. So essentially looking for biocompatible aerosol delivery of exosome nanosuspensions. So transfection and gene modulation are both dose dependent. We look at the amount of cells, so say for CF you'd be targeting airway epithelial cells, you've got 10.5 by 10 to the power of 9, and almost 20 times that number of cells when you look at alveolar epithelial cells in the distal lung. On the plus side, it's estimated you only need to hit about 10% of those epithelial cells in the context of CF to restore CFTR function. So we're going to investigate that critical delivery aspect with these formulations of surface engineered exosomes from an early stage, and that differs in preclinical development methodology, which first develops your vector and then looks for a technology to deliver it. When we look at our microRNA disease targets, a little bit more scientific of approach than throwing a few darts into a list of target genes. So we did a study disease dysregulated microRNAs, and they're validated gene targets and predicted gene targets in the context of the disease we were looking at, which is initially COPD. So it took quite a bit of reading and literature reviews, and you can go down a lot of rabbit holes here, but we've managed to come up with a few novel microRNAs for investigation in COPD. The other side is we can solve delivery-related problems with a better vector, which is also innately therapeutic when you look at an MSC exosome, of huge benefit in inflammatory lung diseases, and then combine that with formulation and proper aerosolized delivery to get the right response. So we're looking at a couple of ATMPs. Exosomes have sometimes been regarded as not as ATMPs, but our bioengineered ones are very much gene therapy ATMPs, and we've got that classification for our initial target indication, which is cystic fibrosis. And it's also the subject of our CALIN proof of concept research strategy, which we'll be doing with Galway and Cardiff University. So, as I said earlier, there's been a long history of failed CF gene therapy. But what's already been shown is exosomes carrying overexpressed CFTR messenger RNA can transfer that into recipient cells and restore functionality. That's been shown in vitro, not with MSCs, but with lung cells. It's also been shown with CHO cells. Some unpublished data says that CFTR mRNAs also could be found in exosomes. So we'll soon figure out if that's the case. MicroRNA 17 is immunomodulatory. It's an anti-inflammatory microRNA that would downregulate interleukin 8 in the lung. It's already been studied and shown to reduce or repress that target gene along with a host of others, such as the complexity of microRNA and messenger RNA interactions. So we want to transform our allogenic MSCs into exosome-producing factories and have them produce exosomes loaded with sequence-engineered CFTR messenger RNA. So we know that there's upregulated microRNAs in cystic fibrosis that actually will repress CFTR expression. So there's about 5 or 6 well-recognized microRNAs there, but we're not going to send in binding sites for those microRNAs. We'll be sending in synthetic, untranslated region constructs that should express widely in most cell types, and also have these exosomes overexpressing microRNA 17. We'll have them surface engineered, as we've already started to do, to penetrate the mucus and deliver these contents into our targeted epithelial cells. And finally, we'll be able to deliver high doses of those to the central lung via suitable aerosol technology, be that one that's marketed or if we have to go to a research bench technology. And there's a second indication. We're looking at COPD. We've submitted grants recently to a couple of agencies in the States for novel discovery geared awards. We have 3 novel microRNAs, and they're chosen as really key regulators of disease pathogenesis in COPD. We want to deliver those within allogeneic and bioengineered MSC exosomes. So these microRNAs, we counted over 70 COPD associated genes that they have been experimentally validated to knock down. So we expect pretty profound effects when we have a look at this in vitro models. We're looking at modulation of pathways. These knockdown of these target genes should upregulate Wnt and Notch pathways and suppress inflammatory cytokine production, have a myriad of effects on the tissue remodeling. There's really sound rationale behind selection of those targets, which we know are validated, and some are just at this stage in silico prediction targets, which may turn out to be false, but they'd be targets that would be really strongly associated with disease pathogenesis. So again, unmodified MSC exosomes already demonstrate efficacy in various animal lung disease models. So anti-inflammatory, regenerative effects, which is a sound base for sending in these overexpressed microRNAs. So our research plans essentially just aim to demonstrate that we can get these produce these exosomes, show that they carry stably, express over a number of passages these microRNAs, and back it up with testing in vitro proof of concept models, so air liquid interface cultures of COPD disease cells, and also with 3D lung organoid models, which will give us small representative cells of the small airways and of the alveoli. So again, we're targeting a slightly different area of the lung. So you're targeting the distal lung with your aerosols as opposed to the central lung in CF. Two pretty different indications. In CF, it only affects about 70,000 people worldwide, but there's hundreds of millions with COPD, and probably the same amount again that have been undiagnosed. So we need a commercially scalable therapeutic to treat these people, and using immortalized allogenic MSCs, we should be able to get nice proof of concept data to show that that is a feasible, if not easy, target. So couldn't dream about doing it without CALIN and the support of some experts in Cardiff University, Arwen Jones, Ada Clayton, Pete Watson, Roisin Dwyer and Frank Barry in Galway, who's my initial point of contact here, and I forgive him for telling me I was speaking on Wednesday today. So really looking forward to working on this. It should be really exciting proof of concept data. I think it's based in quasi-reality, and I'd like to prove it out as being real. And then we work also with some external experts at the Leibniz Institute in Dresden with this surface acoustic wave aerosol delivery technology, and Paul McLaughlin in UCD and Catherine Green in the RCSI, who some of the pioneers in microRNA and lung diseases in general. So that's it. Thank you very much. Yep.
Do we have any questions for Frank?
Just a quick question about the mucus.
Yes.
In the CF patients.
Yeah.
Is it the same composition as normal mucus, but there's just a lot more of it?
No, the pore size is a lot tighter. It's hyperviscous. But if we can prove that we can get these exosomes through that, the mucus in, say, asthma or COPD is not going to be an issue. Some mucus, the mucus barrier is not really going to be, say, when you're targeting emphysema, it's not going to be an issue, but for CF and the chronic bronchitis, it certainly is.
Is there an enzyme that degrades that?
You could pre-treat with, say, Dornase alpha, which is already in the market, or you could use acetylcysteine, and that would have to temporarily pre-treat. What we want to do is just see a worst case scenario and see can we get these exosomes to get through. Now, nanoparticles have been engineered before up to 500 nanometers and shown capacity to diffuse through this mucus with only about a seventh the speed of water. So I think it's feasible. We'll find out soon enough. Yeah.
Thanks. Any other questions? Yes.
For your RNA loadings, are you looking to just do single species RNA, or do you think it's potential to do?
We can do multiple. So we use a commercially available construct, that's already shown the ability to get about over 30 microRNAs, overexpress them and load them in high concentrations to exosomes. The microRNAs, all of them that we're looking at, their presence has been shown in MSC cell populations. So you're not guaranteed a loading of microRNAs. So if we find that we're having trouble loading them in, we have got different targets in mind, but hopefully we'll be able to crack it first time. Yeah.
And any other questions? No? Great. Thank you very much.
Thanks, guys.
So just before I hand back to Frank to close the meeting, I just very, very briefly want to say a thank you to all of the speakers today. It's been a very interesting event, and a thanks to Frank and Mary, Siobhan, and the rest of the Galway team who have organised today's event and delivered it. I think it's been really good at showing how CALIN is bringing companies across Ireland and Wales together with universities to support innovation across the region. Thank you, Frank.
Thank you very much. I know everybody is anxious to leave, so I won't spend more than a very brief moment. The whole point of CALIN is to bring together companies and researchers in research institutes to try and foster a spirit of collaboration and partnership to advance research and development, to accelerate product development, to create employment and growth and all of that. That's exactly what we're trying to achieve, and that's exactly what the objective of today's proceedings was, to have the companies and the researchers all share the podium here and basically all tell their own stories. And you know that there was a series of fascinating stories told today by companies that are relatively early in their life cycle and companies that are much, much later in their life cycle and have products that have been authorized for market, and companies that are somewhere in the middle. I think what was very clear from all of today's presentations is that the future of medicine resides in the technology that we were discussing today. The application of cell and gene therapy, without any doubt, would provide the treatments in the future for the diseases which today are intractable or incurable. The challenge, however, we all can see from today's presentations is still very formidable. The challenges of clinical proof, the manufacturing challenges, the delivery challenges, logistics, all of that, the biological understanding and the science that underpins all of this, it's all still a series of incredible challenges. But nonetheless, I think there's nobody in this room now that would not agree that those challenges are very, very much worthwhile trying to meet. I have to acknowledge the assistance, financial and otherwise, of the CALIN network in events like this. It's been very important. And my last word is to the local team that you saw running around here all during the day, who helped in the management of this, all led by Siobhan Gohan. Thanks to all of them, and thank you all very much for coming, and have a safe trip back home.